All right. Happy New Year, everybody. Just gonna get started and record this review for you guys so you can look back and watch it when you're studying for your exams or your quizzes. I kind of expected no one to show up today because it is New Year's Day, but that's totally fine. I'll still go through this. And then if you have any questions after watching it, you can always email me or Sebastian or Laura too. All right. So I'm going to go over the cell cycle for part 2. Sebastian went over part 1 on Wednesday when he did the Bio285 for 1 review. So if you still need to watch that one, go ahead and watch it. It's on the YouTube channel, and then come watch this one. And then there's my email. If you have any questions, definitely let me know. All right. So we left off with part 1 going through what the cell cycle is and the different checkpoints, specifically talking about interphase and G0 and G1 phase. But now we're going to talk about the end parts of the cell cycle with S, G2, and M phase. So CDK is very important in controlling different events in the cell cycle. Specifically, it suppresses the reassembly of the pre-RCs in the cell cycle. This is important because it makes sure that the DNA replication occurs only once per cycle. You don't want to have DNA replication occurring multiple times per cycle because then you're more likely to wind up with mutations and then you're going to have too much DNA and that's bad. So CDK is important in preventing the DNA replication from occurring more than once. And it does this in 3 different ways. So one way it does it is CDK phosphorylates ORC, which if you remember is the Origin Replication Center, I believe is what it stands for. And when it phosphorylates ORC, this prevents CDC6 from binding. So if you see here, CDK phosphorylates ORC. Once it phosphorylates ORC, CDC6 can no longer bind to ORC and create this complex for replication to occur. But that's not all it does. It also phosphorylates CDC6 itself, so it phosphorylates it here. And when CDC6 is phosphorylated, ubiquitin ligases can now recognize it. And once they recognize CDC6, they can send it to the proteasome for degradation. And it won't be floating around the cell anymore. And then the third way that it prevents re-replication is CDK phosphorylates the MCM27 complexes to initiate nuclear export. So once it phosphorylates this MCM complex, which is involved in replication, then it will be sent out of the nucleus, so it can't participate in replication anymore. An important thing to note, though, with this is that CDK phosphorylates MCM complexes that are not currently replicating. So if you have an MCM complex that's currently involved in DNA replication, CDK cannot phosphorylate it. This only occurs when the MCM is done replicating or not involved. So this is how CDK prevents re-replication, which is super important for the cell. This will also occur in S phase. So in S phase, like we talked about either in class or with Sebastian in Wednesday's review, S phase is when DNA replication occurs. And those were some checkpoints we just talked about with CDK. Well, not checkpoint, but just regulation of the times of replication that occur. And later on in the class, we will talk about the nuts and bolts of DNA replication, but for right now, you don't really need to know how each different protein or molecule that works with it. But for this slide, I just want to go over what cohesin is. So cohesin is a protein ring. It's right here, these green rings that keeps sister chromatids together once they're duplicated during DNA replication. It prevents the chromosomes from segregating away from each other during spindle assembly, which is important because you're going to want the sister chromatids to be together for proper spindle assembly separation. So you get the correct amount of DNA in each daughter cell, which I'll talk about a little bit later. And an important thing to note is that without cohesin loading on these sister chromatids, the spindle assembly checkpoint, which I also will talk about later, will not be able to function properly. And then you'll get problems with your DNA amounts in your daughter cells. So this is before replication. You get your sister chromatids, cohesin is a protein, and it just keeps them together. It's kind of in the name. So now with G2, the regulatory effects of CDK is basically the activation of M phase CDK. So this is occurring in G2 phase, which if you remember comes after S phase. So basically what happens is M phase cyclins are transcribed and translated starting in S phase. So they begin in S phase, but then the levels will start to rise as the cell enters G2 phase because you're going to need more of them. So basically what happens is that CDK, the M phase CDK, is phosphorylated by a kinase called CAC. CAC phosphorylates CDK at the activating position on the molecule, which I have like a visual on the next slide, so it might make more sense then. But also at the same time, CDK is phosphorylated by WEE1, which is an inhibitory kinase. So it phosphorylates CDK at the inhibiting position. So you have two phosphates, one on the activating, one on the inhibiting. So when this happens, the CDK is inactive. It has that phosphorylation at the inhibiting position. It can't do anything with that. So WEE1 keeps MCDK inactive. This is important though, because you need the cell to check for errors in replication during G2. That's the whole regulation of it. Because during G2, you're checking for errors in replication from S phase, because you need to make sure that there's no errors in DNA before you enter M phase for the actual splitting of the cell. And G2 is also another growth phase like G1. So the cell is also growing and getting bigger. But so while the cell is doing this, you need CDK to be inactive, the MCDK to be inactive so that the cell can do it without any errors. If the cell determines that it's good to go, what happens is CDC25, which is a phosphatase, will take off the WEE1 phosphate because that's on the inhibiting position. And now CDK only has a phosphate at the activating position. So MCDK is now active. And then there's this positive feedback loop that causes a rapid rise in the M cyclin CDK complexes, which I'll talk a little bit about in the next slide. So here's what happens. This is the activation of M phase CDK. You start with your mitotic CDK and you get your M cyclin, you make this complex MCDK. However, it's inactive because there's no activating phosphate. So this is before any phosphates are added. Then at the same time, you get both this activating kinase and inhibitory kinase, WEE1 and CAC. So CAC will put a phosphate on this activating position, but WEE1 kind of counteracts that by putting a phosphate on the inhibitory position. Then when the cell is like, okay, we're ready to enter M phase, G2 is all set, CDC25, also known as an activating phosphatase, takes off this WEE1 inhibitory phosphate. So now the only phosphate left on this MCDK complex is this activating phosphate that was put there by CAC. So now this active MCDK can go off and do its thing. And then this is the feedback loop that causes the levels of this complex to rise quickly. So basically CDC25 in a pathway that we don't really know or need to understand for the course of this class, gets partially active in this first pathway. This is a partially active CDC25. It becomes active once the cell signals that it is ready to enter M phase, but it's not fully active. So basically what happens is the partially active CDC25 phosphatase takes off the WEE1 phosphate. Active MCDK can now phosphorylate CDC25 itself. So because now it's active, so it can now phosphorylate this CDC25. So now CDC25 is fully active. It can take off more phosphates from WEE1, the WEE1 position, and lead to more active MCDK, which can then phosphorylate more CDC25. And if you get it, it's this loop that leads to quickly rising levels of MCDK. So that's the positive feedback loop for levels of MCDK complexes. So now we're in M phase. So after once MCDK is activated, the cells are officially in M phase. We have left G2 phase. Everything was good. And now we're in mitosis. I'm sure most of you remember from previous bio classes in high school, M phase consists of prophase, prometaphase, metaphase, anaphase, and telophase and cytokinesis, which are usually clumped together as one step. if you don't remember the main details of each one, that's totally fine. You don't really need to know them for this class. If you take genetics, you will, but for right now, you don't need to know the certain specifics for each phase. But we're going to talk a little bit about anaphase and prophase. Okay. So the reason we needed those MCDK complexes is because MCDK is important for the beginning of M phase. It phosphorylates many proteins during prophase that allow the cycle or the process of mitosis to be initiated. So basically what MCDK does is it phosphorylates 3 different proteins. We have lamins, condensins, and MAPS and motor proteins. So when MCDK phosphorylates lamins, what happens is the lamins can now, it breaks down the nuclear envelope, if you see here. So by breaking down, this is important because you need the nuclear envelope to break down so you have access to the DNA. This is the first step in prophase is the nuclear envelope breakdown. Then MCDK can also phosphorylate condensin. So this is important because once this happens, then the chromosomes condense. We need this for separation of the chromosomes during anaphase. This is just another important step of prophase. And then MCDK can also phosphorylate MAPS and motor proteins, which are really important for spindle assembly. So these are the 3 main things that MCDK does at the beginning of M phase during prophase. This is a little diagram that shows it phosphorylates these different targets. They lead to different things. So M phase now begins with the spindle forms, chromosomes can condense, and the nuclear membrane breakdown. And this is all because of MCDK. So another important thing that we need to go over for you to really get a full understanding of M phase and all its nuts and bolts is sequential phosphorylation. So sequential phosphorylation is basically you get a priming phosphorylation on your substrate, and then the actual kinase can come in and add another phosphate, basically. So sequential phosphorylation is common for regulation. It's a better way for regulation to occur in many pathways. If you remember from the AKT survival pathway, this sequential phosphorylation was common in that. I think it was PIP3 or no, there were 2 different proteins, protein kinase 1 that would phosphorylate AKT and all that. So basically for this scenario, CDK is known as a priming kinase. So if you look here, you have your substrate. It adds this phosphate here to the substrate, which is known as priming phosphorylation. And it does this for an enzyme called PLK1 kinase. So PLK1 is an important enzyme for the G2 M phase transition and spindle assembly, but it can't phosphorylate its substrate unless CDK also phosphorylates it. So CDK comes in as this priming phosphorylation. This is an inactive PLK. This is just its structure. It then can phosphorylate the substrate also because now it has this phosphate. So basically what it does is it's inactive because it needs to bind to this phosphate that CDK put there. So without CDK, it wouldn't be able to phosphorylate the substrate because if you look here, it has a little spot for this phosphate put there by CDK. So what happens is this PBD domain of the inactive PLK can now bind to this phosphate on the substrate. I'll do here instead. So now it can bind to the phosphate on the substrate. It's now active once it binds, and then the kinase domain of PLK1 can then add a phosphate to the substrate, which is the actual phosphorylation that occurs. So the only real reason the CDK phosphate's put here is so that the inactive PLK1 can bind to it, become active, and then add its phosphate and do the actual phosphorylation of the substrate. So that's sequential phosphorylation. So now that we're in M phase, an important part of regulation is the spindle assembly. Spindle assembly is super important. This is what grabs onto the sister chromatids, pulls them apart to the different poles of the cell, and when the cell splits, you now have equal amounts of DNA. So spindle assembly begins during prophase. Like I mentioned, it's when CDK phosphorylates the MAPS and motor proteins. So basically, when spindle assembly begins, the cell has to disassemble the entire existing microtubule network and then reassemble the tubulin into a spindle structure. So if you remember from the microtubule lectures, microtubules are made of tubulin, and it has to do this reassembly into the certain spindle structure, but it first has to disassemble the entire existing network. So in order to do this, the cell uses MAPs and motor proteins to disassemble and then reassemble. So this is the structure of them. They extend from the spindle pores and then will overlap in the center. And then they're held together by these proteins. You can see here these black dots, which stabilize the spindle. So see how they extend out from the pores towards the center and then overlap. And they're held there by these proteins, which is important because you need to stabilize the spindle in order for it to work properly. So there are different types of microtubules. You have aster microtubules, kinetochore microtubules, and interpolar microtubules. I'll start with aster microtubules because they're the outermost microtubules. They're always out on the edges. So basically they attach to the spindle pores. They attach the spindle poles to the inner portion of the cell membrane. So here's your spindle pole, and the aster microtubules, which are here in green, extend out to, you have this cell, it's not in this picture, but you have the cell membrane here just around it. And that's where the aster microtubules attach to, the inner portion of it. They don't go outside of the cell. Then the interpolar microtubules, which are here in like pink, they extend from the pole to the middle. And these are the ones that overlap and are held together by these proteins. They basically stabilize the spindle. That's their main job. And then lastly, in blue, we have kinetochore microtubules, which come from the pole and they attach to the kinetochore proteins complexes that are on the actual sister chromatid. So these little red marks are the kinetochores, and then the blue lines are the kinetochore microtubules. And basically they extend from the pole and attach to the kinetochore proteins on the sister chromatid. And these are important for when the cell separates, or when the chromatid separates and the cell divides. So these are your three different types of microtubules. They're important for they have different functions, and they're important mainly for spindle assembly and separation of the sister chromatids during cell separation. So spindle assembly begins with the kinetochores. Kinetochores are large protein structures that are on the centromere of each sister chromatid. So this is the centromere region, which is an actual part of the sister chromatids. And then you get this kinetochore, which is a protein, this red part on the centromere region. So it's not a part of the sister chromatid, it's just on it. And then these green lines are the kinetochore microtubules that attach to the kinetochore. So just for a reminder, because some people tend to get confused, kinetochore microtubules and kinetochore proteins are completely different structures. So when you're referring to, if you refer to something as just a kinetochore, you're usually referring to the protein structure on the sister chromatid. If you're ever talking about the microtubules, you should always really say kinetochore microtubules. Otherwise you might get them mixed up. And then this is just a chromosome. So that's just a quick side note, because some people may get confused with that. So now building the spindle. This is done in a push-pull mechanism. So basically what happens is opposing forces keep the spindle stable, this like pushing and pulling, and keeps the spindle poles at a certain distance away from each other, because you don't want them to get too close. You also don't want them to get too far either. And this contributes to the forces that pull the sister chromatids apart during anaphase. So dynein is a minus end directed motor protein. So if you remember, microtubules have a plus end and a minus end. And during the spindle assembly, basically what happens is dynein binds to the aster microtubules. So remember, those were the outermost microtubules, and the cell cortex. So as it walks towards the minus end of the microtubule, it creates a pull force. So dynein is a minus end directed motor protein that creates a pull. So it starts near the outside of the spindle and goes towards the inside or the middle. Now we have EG5, which is a plus end directed motor protein. So it starts in the overlapping interpolar microtubules in the middle of the spindle and walks towards the plus end, which creates a push mechanism. So It starts in the middle and goes towards the outside. So dynein is a minus N directed motor protein, creates the pull. EG5 is a plus N directed motor protein and creates the push. So these two proteins work to build the spindle by creating this push-pull mechanism. And then just a little side note that you should know, monostrol is an EG5 inhibitor. And basically what happens is it creates this monopolar spindle because the only force you have is this inward pulling by dynein. There's no push. So what happens is when dynein is pulling, but there's no push, the spindle will collapse. So this just shows how important EG5 and dynein are. Monostrol, I don't know its implications and when you would use it, but I'm sure you could look that up. I don't know if it's medication or just something, but anyways, the spindle will collapse if you have only one of the two push and pull. So those are just the two things to remember. Dynein pulls, EG5 pushes. Dynein's a minus end, EG5's a plus end. So once again, there's another checkpoint because we have so many checkpoints throughout the cell cycle. Otherwise, we'd have crazy mutant cells just replicating like crazy. We don't want that. So we have another spindle assembly checkpoint. This is where the cell transitions from metaphase to anaphase. So anaphase is when the sister chromatids actually separate. Metaphase is when you're building this metaphase plate, and this is where the sister chromatids line up in the middle of the cell. So the two proteins we have here are MAD and BUB. They are two checkpoint proteins. What they do is they sense tension in the chromatid pairs. When like the cell is like ready to pull them apart, they sense tension in the microtubules that are ready to like pull the sister chromatids apart. So when they sense this tension, what happens is CDC20 is released from sequestration. And then when CDC20 is released, it binds to APCC, which is just APC for short. And APC is a ubiquitin ligase, so it will tag something with ubiquitin and send it to a proteasome for degradation. So what happens is now we have this complex of CDC20 and APC, and it ubiquinates a protein called securin, which is up here in brown. This is an inhibitory protein that inhibits this blue protein called separase. So this is important. You don't want separase to constantly be active, otherwise you would get separated sister chromatids too early and all that. So it's a very timed checkpoint. It basically is when the cell has everything lined up, it's ready to separate the chromatids. MAD and BUB initiate it, and then CDC20 ubiquitinates securin. So it adds this tag to securin, and then as you see, it goes to the proteasome for degradation. So now separase is now active. Oops. So when separase is active, it's a protease, which means it cleaves cohesin, which are those protein rings around each around the sister chromatid pairs to keep them together so they don't split too early. So separase, if you think of the name, kind of easy to remember, separase separates the sister chromatids. So what it does is it cuts these bonds between cohesin, the cohesin rings. So now the sister chromatids can separate. So if you look here, so now these are dissociated cohesins. They just go off. They're not important. And this is the big trend. This is what signals the transition from metaphase to anaphase. So metaphase is when you still have these cohesin rings. As soon as they're cut and sister chromatids are separated, you now are in anaphase. And since there was already tension from the kinetochore microtubules, once the sister chromatids are free, the kinetochore microtubules can now pull them to the opposite poles. So this is this checkpoint. This is a regulatory mechanism to prevent the sister chromatids from being separated too early, and also signals the transition from metaphase to anaphase. There's two types of anaphase. There's anaphase A and anaphase B. Some cells do both at the same time. Some will do one and then the other. I'm not entirely sure the specifics, but you just need to know that anaphase A is when the chromosomes are pulled apart by shortening kinetochore microtubules. So the chromosomes, so basically the kinetochore microtubules are attached to the sister chromatid kinetochores, shorten. So they pull the sister chromatids apart. And the chromosome is walked towards the spindle pole by a -N directed motor, like dynein. So it creates this force that moves the chromosomes towards their spindle pole because the kinetochore microtubule is shortened. So if you see here, this is one kinetochore microtubule, and this is it shortened. So it just pulls them apart. So that's anaphase A. The chromosomes are pulled poleward by shortening kinetochore microtubules. And then anaphase B is the poles are pushed and pulled apart. So this is basically to create a sliding force between the interpolar microtubules, which are these ones that overlap, if you remember, from opposite poles to push the poles apart. And then the pulling force acts directly on the poles to move them apart. So this is 1 and then 2. So they're moving, the interpolar microtubules are moved this way so that the poles will move this way. Basically, this is to lengthen the spindle and push and pull the poles apart. Motors like dynein on the aster microtubules, which are these ones out here, will pull the spindle poles apart, and then EG5 motors located on these interpolar microtubules will push the spindle polars apart. So it's that push-pull mechanism that we talked about before, so that the interpolar microtubules can lengthen. This just creates like a bigger spindle to move the chromosomes or the chromatids farther apart. So some cells do these at the same time, these two mechanisms. Some cells do one and then the other. You don't really need to know the specifics, but that's just anaphase A and anaphase B and how they differ. And finally, cytokinesis and telophase, pretty much, is just the separation of the cell, the two daughter cells. There's this contractile ring, which is somewhat, I think this is the ring actually. Yeah, so this is the contractile ring. So it's made of actin and it's in the center of the cell. And what happens is it constricts until these two, it creates a furrow, like this cleavage furrow. And then basically what happens is it keeps going until these two cells separate. And then just a side note, this isn't really connected to cytokinesis, but it's something you should know, is that at the beginning of anaphase, M cyclin levels drop. If you look here, this is the cyclin expression levels during the cell cycle based on, the phase they're in. So you have your G1 cyclin increases pretty consistently and then decreases also pretty consistently. Like we talked about in S phase and M phase, it increases pretty quickly. But then we're talking specifically about N cyclin here, it drops off very quickly during anaphase because during anaphase, once you split your sister chromatids, you want the nuclear envelope to reform, you want the spindle to disassemble, and you want the chromosomes to decondense. You don't need these in prophase anymore. So this is why at the beginning of anaphase, M cyclin levels drop. And this has also occurred, if you remember talking about the spindle assembly checkpoint. This APC complex to CDC20, it targets M cyclin. So this is what causes these levels to drop very quickly during anaphase or at the beginning of anaphase, because you don't want M cyclin levels around, because then the nuclear envelope won't reform, the spindle will still be there, and the chromosomes won't decondense, which is not the proper conditions you want for your new cells to be in. You want the nuclear envelope to reform. You don't want a spindle around, and you want your chromosomes to be not condensed. You want them to decondense. So these are some questions I had. One is just based on what we talked about, the nuclear membrane or nuclear envelope, whatever. And then 2 is just to talk more about proteins, just, to get your mind going back to the first stuff we talked about in class. So if you do them on your own and think you know the answers, great. I'll pause for a minute, let you guys answer them on your own, or you can pause your video too, and then I'll go over the answers. So number 1, the nuclear membrane is absent in which of the following phases of mitosis? A, anaphase, B, telophase, and C, metaphase. Should also probably let you know that the answer could be more than one in this one. All right. So the answer for this question is, what is the answer? Okay. The answer is anaphase and metaphase, because the nuclear membrane is degraded at the end of prophase. Remember, this is when CDK phosphorylates lamins, which cause the degradation of the nuclear envelope. So the nuclear membrane or envelope, whatever, is degraded at the end of prophase so that the chromosomes can be appropriately separated into the daughter cells. So it doesn't reform until anaphase. So technically, it doesn't reform until telophase and cytokinesis. So remember what I talked about here, anaphase, at the beginning of anaphase is when these levels drop, but they don't actually reform until cytokinesis, and like all of this happens during cytokinesis. So the nuclear membrane is absent in anaphase because this is when the levels are dropping. Metaphase, it's definitely still absent because levels of M-cyclin haven't decreased yet. But the nuclear envelope, it reforms in telophase or cytokinesis kind of together. And that's why A and C are the correct answers. And then number 2, which of the following best describes the secondary structure of a protein? A, various folded polypeptide chains joining together to form a larger unit. B, the amino acid sequence of the chain. C, the polypeptide chain folding upon itself due to the hydrophilic and hydrophobic interactions. Or D, peptide bonds hydrogen bonding to one another to create a sheet-like structure. So the answer here is D, because if you look at the other answers, various folded polypeptide chains, that's more than one polypeptide chain. So you know automatically that this is talking about quaternary structure. So we know that's not secondary. The amino acid sequence of the chain, that's primary, that's your original polypeptide. C is the polypeptide chain folding upon itself due to hydrophilic and hydrophobic interactions. So this is your R group interactions. And if you remember, secondary structure has nothing to do with R groups. It is only the backbone, and it is only hydrogen bonds between the backbone. It's not any hydrophilic, hydrophobic interactions. That's tertiary structure. So that just leaves us with D, which is correct. Peptide bonds, hydrogen bonding to one another to create a sheet-like structure. This is secondary structure. And that's all I have. So if you guys have any questions after watching this, you can always email me. I hope this was helpful, and I hope you guys have a great start to your new year.